I sent it to her.
As she will join, she will show here Mania Ajmal. You have to just click and make it host on stage. No further currently moved link to all communities as well.
Sure. Kindly turn off the sound right now. We are gonna start. Assalamu alaikum and a warm welcome to everyone. On behalf of Vitanova International Alliance for Science, I am very pleased to welcome all the participants, distinguished guests, faculty members, and aspiring researchers to the Bioinformatics and Molecular Docking Online Internship Program 2026. We are truly excited to have participants joining us from different types of institutes, backgrounds, as well as countries, united by a common passion for science, research, and innovation. This internship has been carefully designed to provide both theoretical knowledge and practical insight into the rapidly growing field of bioinformatics and molecular docking. Before we begin, I would like to share an important announcement regarding attendance. A Google Form link will be shared in the live chat during this session. All participants are requested to fill out the form carefully to mark their attendance for the day 3. As mentioned in the internship guideline, attendance is mandatory for certification. So please ensure that you submit the form before the session ends. Now it is my great honor to invite a visionary speaker which took two sessions before as well to guide you as well as gave you practical knowledge. Our estimated speaker, Ms. Mani Hajmal. As you all know about her already, I'm not gonna give the introduction again. So, without any further delay, let's go to her and give her the floor so that you guys get the basic knowledge about the blast in bioinformatics from theory to hands-on application. Miss Maniha, the floor is yours. Kindly go on.
Thank you so much, Noor. And good day, everyone. Hope all of you are doing good. So, yesterday we talked about some of the databases NCPI, Uniprot, PDB, KEGG. And today we are going to go deep into the tool that sits at the center of all the sequence databases, that is BLAST. Why do you use BLAST? So, if you look at the sequence database, BLAST is the most used tool in all of the bioinformatics.
Miss, you were not audible.
If you take a sequence and you search it against all the entire known sequence universe, hundreds of thousands of base pairs deposited by researchers over 4 decades, and within seconds you will find whether anyone has ever seen that sequence before or not. And if they have, BLAST tells you what it is, what organism it comes from, how similar it is to your sequence, and how confident you should be in that match. So BLAST, which is short for Basic Local Alignment Search Tool, it's just not a tool. It is the most used tools in all of the bioinformatics, and it has been cited in scientific papers more times than almost any of the other methods in biological research. And understanding it properly, we not just know how to click the button and how to do it, but understanding the algorithm and how it works and what the numbers in the output actually means is one of the most important skills you can develop as a bioinformatician. So that is what we are going to do today. The first half will be theory, and the second half is going to be the hands on training. And by the end of the session, I hope all of you will not be only able to run Blast, you will also understand what is beneath it and what the results look like and why do they look like that. So let's start officially. So can you move on the next slide, please? Okay, thank you. So, first of all, we discuss why BLAST matters. I've already told you that it is the one of the most widely used bioinformatics tool, and it has the ability to identify an unknown sequence within seconds. So it is one of the most widely used bioinformatics tool. So before we talk about BLAST, we need to understand the problem as well and why was BLAST made. Can you move to the next slide, please? Okay, so the problem that BLAST was made to solve was that how do you find sequence in a database that are similar to a query sequence you care about? For example, if you have a query sequence and you want to know what does it looks like in other databases and whether it exists or not. So this was the fundamental problem that BLAST wanted to solve. At first that sounded simple, but think about the scale. The NCBI nucleotide database contains hundreds of billions of databases and base pairs. A typical gene is maybe 3000 base pair long, and if you want to compare your 3000 base pair query to every single sequence in that database, one position at a time, so what approach would you do? I mean, if you apply a brute force approach, you would be waiting for hours, potentially days for results on a single search. And it's not just a speed problem, it is also a biology problem. When two organisms share a common ancestor, their genes diverge over evolutionary time. Mutations may accumulate, nucleotides may change, insertions and deletions may also happen. So, two sequences that have genuine evolutionary relationship to genuinely homologous genes might not be identical. They might not even be very similar at the nucleotide level. So, how do you find a meaningful biological relationship in a sea of partial imperfect similarity? This is a challenge that sequence BLAST is going to solve, and BLAST is the answer that has dominated the field since 1990. So now we're going to talk how sequence alignment work. Can we move on to the next slide? Next one, please. Okay, thank you. So to understand BLAST, you first need to understand sequence alignment. That is the foundational concept that underlines the whole thing. Sequence alignment is actually the process of arranging two or more sequences so that similar characters are placed in the same column. So it allows you to see where they match, where they differ, and where one has insertions or deletions relative to the other one. So there are two main types of alignment, sequence alignment. One is global alignment, one is local alignment. Global alignment tries to align two sequences from one end to the other end, like across their entire length. And the classic algorithm for this is Needleman-Wentz that was published in 1970. And global alignment is appropriate when you are comparing two sequences that are approximately the same length and they are expected to be similar throughout from one end to the other end. Like it is like comparing two orthologous genes from closely related species, for example. Whereas on the other hand, local alignment finds the best matching region between two sequences, but it does not try to align them end to end. And the algorithm that is used for local alignment is Smith Waterman, that was published in 1981. Local alignment is appropriate and far more powerful for database searching when you expect only part of your query to match a part of the database sequence. For example, if your query sequence and you want to match a small sequence in a sea of databases, so you use local alignment rather than global alignment. So local alignment finds that domain even if everything around it is different. So BLAST also uses local alignment. This is the key design choice because it means BLAST can find a conserved kinase domain in the middle of otherwise unrelated protein or a conserved exon in a sea of divergent intronic sequence. So that flexibility is one of the reasons it is very powerful. Can you move on the next slide? Okay, so Any realistic alignment between two related sequences will also contain gaps, and gaps are the places where one sequence has an insertion or deletion relative to the other. These gaps are correspond to insertions or deletions that are called as indels that occur during evolution. You all know what mutations are. In alignment algorithm, gaps are penalized because they represent evolutionary events that should not be introduced previously, and the penalty structure typically has two components: a gap opening penalty for introducing a new gap, and the gap extension penalty for making an existing gap longer. So, opening a gap is penalized more heavily than extended one that already exists. Because a single evolutionary deletion event can remove many consecutive nucleotides at once, so a long gap is not necessary evidence of many independent events. So the specific gap penalizes that you affect that you use affect alignment quality significantly. So, if we talk about BLAST algorithm, and I'm going to tell you how BLAST actually works. So, the key insight behind BLAST is that it does not try to compute a full Smith Waterman local alignment between your query and the sequence in the database. That would be far too slow. Instead, it uses a heuristic, a fast approximation that is actually a bit of trade off. It is a trade off between sensitivity and speed. So the BLAST algorithm has three stages, and understanding these three stages is the key to understand both the power and the limitations of BLAST as well. So if we talk about the three stages, stage one is seeding. And BLAST starts by breaking whatever query sequence you provide. It breaks your query sequence into short overlapping words, which are called as schemers or seeds that could be of any specific length. BLAST typically use a schemer of 11. That one is the default. You can set it yourself as well, but the BLAST n uses a default size of 11, meaning BLAST takes every consecutive 11 nucleotide stretch in your query, whatever query you provide. It will search it over a schema of 11 nucleotides. And for protein BLAST, the default is 6. And BLAST considers not just the exact word matches, but also similar words based on a scoring matrix. Okay. So BLAST then scans the databases looking for exact matches to these seed words, and this is fast because it can use pre-built lookup tables. The database is indexed, so finding exact word matches does not require reading every character in a database. It jumps directly to the location where matching words occur. So the logic here is that if two sequences are significantly similar across a region, they are almost certainly going to share at least one short word exact match within that region. So finding those seed matches is a fast way to identify candidate regions for further investigation without having to examine every position in the entire database. Then it moves on to the stage two, that is extension. Can you move on? Okay, next slide. Thank you. Once BLAST has found a seed match, a short exact word match between the query and the database sequence, it enters the extension stage. It starts from the seed position, and BLAST extends the alignment in both directions, left direction and right direction from the seed, and it adds one residue at a time, and scoring keeps on going as the alignment is going on, and the extension stops when the alignment score drops below a threshold relative to the best score we've seen so far. And this threshold is called X, that is called the drop off parameter. And if the cumulative score falls below the threshold, the best score that is achieved during extension, BLAST stops at that point. This prevents the alignment from wandering into unrelated sequence regions that happen to follow a good seed match. This process produces what are called the high scoring segment pairs, and an HSP is a pair of aligned segments, one from the query, one from the database sequence with no gaps, whose alignment scores exceed a threshold, and we set a threshold before as well. So in the original BLAST algorithm, HSPs were ungapped. There were no insertions or no deletions allowed during extension, like in the modern BLAST Plus, that goes on to perform gapped extension on the best HSPs using a more sensitive algorithm that can introduce gaps. And this gapped extension is more expensive computationally, but it is much more biologically realistic as well. And the final stage is evaluation, and it determines which HSPs are statistically significant. This is where the E value comes in, and we're gonna study the E value later on as well. And I told you about it a bit about it yesterday as well. Can we move on the next slide? Okay, so BLAST scores each HSP that it forms, and it converts the score to a statistical measure of significance and returns that results rank by significance. And database hits below a significant threshold, typically an E value that should be less than, say, 0.01 or 0.05, depending upon the analysis, are reported as output. So next we move on to the scoring metrics. Okay, thank you. So we know that every alignment needs a way to score matches and mismatches. That is a numerical system that says how good it is to align one residue with another. For nucleotide sequences, this is relatively simple. As I've already told you, that matching nucleotides get a positive score. If it's a mismatch, it will get a penalty. But for protein sequences, it is considerably more interesting and it's a bit complicated, as we all know that all amino acid substitutions are not equally likely or consequential. For example, leucine and isoleucine are chemically very similar. Both are non-polar, both are aliphatic, both are hydrophobic. So substituting one for the other is relatively common in evolution, and it has also little functional effect. But a substitution of glutamate for arginine, on the other hand, it involves a dramatic change in the charge, and it is far rarer and usually more disruptive. So, scoring metrics encode these evolutionary likelihoods as numbers. For the proteins one, there are two. One is PAM matrices and other is Blossom matrices. PAM means point accepted mutation, and these are the matrices that were developed by Margaret Dayhoff in 1970s. One PAM unit represents the evolutionary distance at which 1% of amino acid positions have changed. And these matrices are built by examining closely related protein sequences, counting which amino acid substitution actually occur, and extrapolating to longer evolutionary distances mathematically. So PAM1 describes 1% divergence. PAM 2.5 describes the expected substitution frequencies at a point where on average each position has been substituted 2.5 times. And there are different variants of PAM. PAM 250 is calculated from PAM 1, not directly observed, which is one of their limitation. These are some of the very deep concepts, but I would like to explain them a little bit easy so you might understand. So they have actually gave a scoring matrix where each amino acid changing into another amino acid had a point, a score. For example, changing leucine into isoleucine does not have so much of biological significance, so it is a low score. Whereas changing of leucine into arginine, it's a bit complicated, so it gets a high score. So this is how the scoring is done in case of proteins. And for Blossom matrices, that is short for block substitution matrix, and these were developed by Hanikoff in 1992, and they are what BLAST uses by default. The key differences from PAM matrices is that Blossom matrices are derived directly from observed substitution in conserved protein marks. It does not look at the amino acid to amino acid in complete protein, but it looks at the domains, and it compares the domains with other domains, and it captures the change in amino acids in the domains in the protein families rather than extrapolated from a single evolutionary step. And the number in every matrix name refers to the percent identity hold used to cluster the sequences before counting the substitution. And Blossom 62 is the default for BLAST P, and it means a 62% identity, and we consider it as a threshold for BLAST P. And another rule is that higher blossom number means more similar sequences were used to build the matrix, making it better for close homologs. So if you're searching for a very distant relatives, you should use a lower blossom number, like you can use blossom 45, you can use blossom 32. So there are different variants of blossom as well. Can we move on to the next slide? Next slide, please. Okay, so now we're going to discuss about e-value. And whenever you're going to look at blast, you will look at the e-value at the first. So you should pay attention to this part. So e-value is one of the concept that is most commonly misunderstood as well. So let me tell you what e-value is, what it is not, and how to use it correctly. So the e-value, or it is called an expect value, it is the number of alignments with a score equal to or better than this one that you would expect to find by random chance alone when searching a database of this size. Okay, so it is a number of alignments you would expect by chance in a database of this size. For example, an e-value of 1 means that if you search a random database of the same size with a random query, you would expect to find one alignment this good by chance, and that is not significant hit at all. It could easily be a coincidence. But an e-value of 0.01 means that searching a random database of the size, you would expect to find this alignment by chance once in thousand searches. That is starting to look a bit meaningful. And e power minus 50 means that you would need to run this search 10 to the power of 50 times against a random database before you would expect to see this match by chance. So this is extraordinarily significant. If you see an e value like that, that means that the match is real. So the e value depends on three things: the alignment score, the length of the query, and the size of the database. A longer query is more likely to produce spurious matches by chance, so the same alignment score will give a higher e-value for a longer query. And a larger database will have more sequences, so the same alignment score will give a higher e-value in a large database. And this is important. If you search different databases, the same hit can have different e-values depending on the database size. And the alignment score itself depends on the number of matching residues, the number of mismatches, and the number of length of gaps weighted by scoring metrics and gap penalties. Okay, so common threshold. What is the common thresholds then that we use for e-value and how to apply them? The conventional threshold for significance in BLAST is e-value less than 0.01 or 10 raised power minus 3, but this is a convention, not a biological law. The appropriate threshold can depend on your question. And for identifying a gene in a closely related species, you might require e-value less than e to power minus 20. For detecting very distant homologs, for example, if you want to detect ancient conserved domains, then you want to consider low values such as e power minus 0.01 or zero point 0.1 as worth investigating provided the alignment makes biological sense. Okay, so there are three common mistakes that e-value people do. First of all, e-value is not a probability. E-value is not the same as the p-value. For example, e-value equal to 0.01 does not mean 0.1% chance of being wrong. Probability is different from e-value. And second mistake people do is that using the same e-value threshold for all searches regardless of the query length or your database size. So e-value also depends on the database size and also depends on the query length. And mistake number three is they accept a low e value without looking at the alignment as well. A low e value with only 30% coverage or 40% percentage identity, it is a very different result with a low e value and with 95% coverage and 95% identity. So always look at the percentage identity and query coverage as well, and e-value as well when you are considering the data, the results from BLAST. So there are various variants of BLAST, and you should know which one to use and when to use those. Can we move on to the next slide? Okay, thank you. So BLAST is actually not one tool, it is a family of tools, each designed for a specific type of query against a specific type of database. And choosing the wrong variant is one of the most common mistake beginners make. So let me be very explicit about it. So there are different variants of BLAST. The number one is BLAST N, that is nucleotide to nucleotide. BLAST N searches a nucleotide query against a nucleotide database. So, for example, if you have a DNA or an RNA sequence and you want to find similar DNA or RNA sequences in a database, so you would use BLAST N. And for example, what are the DNA or RNA databases we studied yesterday? GenBank, we have RevSeq. So it searches across those databases. So its use cases are, for example, if you have identified a gene from a sequencing read and you want to find where your PCR product maps in the genome, you want to check for contamination, you want to identify a pathogen from a metagenomic sample. So that is when you use BLAST N. Then we have BLAST-P, that is protein versus protein. BLAST-P searches a protein query against a protein database, and you will use it when you have an amino acid sequence and you want to find similar proteins. So this is the most sensitive BLAST variant for finding distant homologs because amino acid sequences are far more conserved than the nucleotide sequences that encode them because the genetic sequence is degenerate, so many nucleotide changes do not change the amino acid. And its use case is characterizing a new protein, finding the human ortholog of a mouse protein, identifying the function of an uncharacterized sequence. And then we have BLAST X, that is translated nucleotide versus protein. So BLASTX actually translates your nucleotide query in all the six reading frames, three forward, three reverse, and searches each translation against a protein database. When you give a nucleotide and you want to search it across the protein database, then you are going to use BLASTX. What it does is that it takes the three amino acid that is short for one codon and it looks which amino acid it encodes and then it searches for the database, the protein database. And this is particularly useful when you have a genomic or a transcript sequence and you want to find what protein it encodes, or when the nucleotide sequences are too divergent to find with BLAST N, but the proteins are still related. And its use case may include when you are annotating a newly sequenced genes and you want to find the proteins homologous to that sequence. Okay, then we have T-BLAST N, that is protein versus nucleotide database. So T-BLAST N does the reverse. It takes a protein query and it searches it against a nucleotide database that it translates on the fly. Use it when you want to find the genomic location of a known protein, or when you suspect a gene exists in a genome that has not been annotated at the protein level, and its use case include finding the coding sequence for a known protein in a genome assembly. And then we have T-BLAST X, that is translated versus translated. It means that both the query and the database in all the six reading frames and compares them at the protein level. It is the most sensitive but the slowest variant, and you use it when you're comparing distantly related nucleotide sequences at the amino acid level. It is the translated sequence, not the original nucleotide sequence. Okay, so next we move to the BLAST interface and the parameters. Can we move on to the next slide? Okay, thank you. So before we run the live demos, let me walk you through the key parameters you will see on the NCBI BLAST interface. And understanding what each parameter does means that you can adjust searches intelligently and you adjust them yourself rather than accept the default. So database selection is the first part. The database you search against is one of the most important choices. The main options on NCBI are NR to NT, the non-redundant nucleotide or protein databases which contain sequences from GenBank, RevSeq, and other sources. And it is the most comprehensive. RevSeq, it's the curated and non-redundant reference sequences, more reliable and faster to search. And then we have UniProt and SwissProt, manually reviewed proteins only. and the organism-specific databases for common model organisms. Then we have word size. As we discussed in the theory section, word size controls the length of seeds as well that the blast uses. If you're giving larger word size, it is faster but it is less sensitive. And if we are giving smaller word size, it is slower but it is more sensitive and it finds more distant homologs. So NCBI blast defaults are 11 and 6 for protein. And if you're not finding results you expect, you can reduce the word size. And if your search is taking you too long, you can even increase it. Then we have the e-value threshold. So this sets the maximum e-value of hits to the report and default is 0.05. For most of the searches you want to tighten this to e power minus 5 or e power 10. So for searches where you expect to find only very distant homolog, you might loosen it up to 1 or even 10. But be prepared to examine alignments carefully and not over-interpret the weak hits. Then we have scoring metrics and gap penalties. As compared in the theory section, Blossom 62 is a default and it is usually appropriate, but you can adjust this yourself as well. Then we have an organism filter in entrance query. It actually restricts your search to specific organisms or groups. For example, typing Homo sapiens search only for Homo sapiens, and typing bacteria restricts to bacteria. This dramatically speeds up the searches and reduces noise when you know which organisms are relevant. But if you do not know which organism your sequence belongs to, you can leave it empty. You can also set the maximum target sequences. For example, if you want 100, the default is 100, you can set it yourself as well. And if you're doing phylogenetic analysis, you may want a broad sampling of related sequences. You may increase it as well. So now let's move on to the live session, and I would need to share my screen for that. So give me a moment. Okay, so I hope my screen is visible to all of you.
Yes, it is peaceful.
Okay, thank you so much. So today we are gonna do the practical demonstration of Blast and how to use it and when to use it and what are the different variants of Blast. So I'm gonna, explain it a little bit more. So we have, four different types of Blast that you can use, but you mostly use nucleotide blast and protein blast until and unless you are doing extensive research, then you may need to use blast X and T blast N as well. I have explained the difference between all the different variants of blast. So this is the interface that blast has when we are going to do a blast search. So first of all, we're going to do the nucleotide blast and then we're going to do the protein blast. So you're going to open the nucleotide blast and you're going to get the interface like this. So if you're doing your own research, you have to put your FASTA sequence here yourself. This is the first part of BLAST, that is the enter query sequence, and you can shift your the variants of BLAST from here as well. For example, I am on BLAST N right now. If I want to shift to BLAST B, I can do it from here. If I want to shift it from BLAST X, I can do it from here. So first of all, I am going to select the BLAST N that I'm going to do right now. So if you have your own sequence, you can paste it. If you have a file, you can choose the file from here. But if you are do not have, for example, right now I am just doing a demo, and if you want to just learn, you can go to NCBI and you can take any sequence. For example, I am going to NCBI. I told you yesterday how to get a sequence from NCBI. I'm going to select nucleotide database because I want the nucleotide sequence, and I'm gonna search any kind of, for example, I am going to search BRACA1 and I'm gonna get the sequence for this. Okay, so let's take this sequence. This is going to be very large, so let's take a slightly smaller one. Let's search pseudomonas because it's gonna have a little, slightly smaller. That would not take too much time, but because the larger the sample, the more it is going to take you the time to run the faster sequence. So let's search pseudomonas. Okay, so this also has a large amount of base pairs. So let's do this one. We're gonna open the faster sequence and we are simply gonna copy the FASTA sequence from here and paste it in our nucleotide blast. Okay, so this is the first step. So query subrange means that from which base pair to which base pair you want to do it. But if we set it default, it means that it will search from first to the last base pair. So that's we want to do. So job title, you can give any job title. If I click here, it will automatically give the name of this species that the sequence is from. But if you want to give any other title, you can give any other title as well. So this is the second part of BLAST that lets you select the organism or the databases you want to select you search the nucleotide against. So we set it as standard databases because it is going to search it across the nucleotide databases. Now we are going blast n, so nucleotide will be searched across the nucleotide databases, so we are not going to change it. But if you want to do something else, you want to check it against some specific databases, you can select from here. But we're gonna do it, just a second, yeah, the default one. Okay, so here if you know which organism this is from, for example, this is from Pseudomonas, you can write Pseudomonas here. That will make the search easy. But if you do not want to add, that's also totally okay. It will take a little bit time, but your results will be the same one. And then we have the program selection here. we always do similar sequences or megablast. Dissimilar sequences is when you want to find the diverged nucleotides, or you want to find, for example, for my Pseudomonas pyrrhida, I want to see the sequences that are the most dissimilar to that one. But I want to find the ones that are highly similar sequences to my sequence. So I'm going to select highly similar sequence, and this is the option that allows you to show the results in a new window, or you, if you want to display the results in the same window, then you're not gonna tick it. But if you want to display the results in a new window, you're gonna tick it and simply run blast. So this is going to take a few minutes. You will be redirected to this page and it will take about 15 to 20 seconds. So, as you all know, this was a real sequence fragment. So, we actually did not have idea that what Blast is about to do. So we just put it here and we hit Blast. So these are the results. So if we see here, this was the job title that we had already given. We use the program blast and the database it search it, description, a little bit description, and these are all the results. So here we can select our e value to, we can put our thresholds ourselves as well, but the defaults are already given in the results, so we are gonna see them in a few moments. But we can set the percentage identity, query coverage, and e value ourselves as well. But for these are the kind of results that we get from Blast. If you see, there are four types of results that you can see in Blast. So first one is description based. So in this you will get all the species that were similar to the sequence you entered. So if you see the first one, it has 100% query coverage. It is a e-value of 0. It is a percentage identity of 100%. And the second one as well. So it means that we have the exact sequence that we put in the search bar, and the database it searched, it had 100% percentage identity, 100% query coverage, and 0 e-value. So what does all this mean? We already discussed that e value is the significant value that it happened by chance, and it should be very low for significant results. Query coverage is that, for example, if we have a length of 12,000 to 68, it should match the 12,000 to 68. It's the query coverage, it's the length it covers across the database. If the length matches, then query coverage is going to be 100%. And percentage identity means that the sequence we gave and the sequence the database searched, it is 100% similar. Each amino acid matches each other amino acid in the sequence that the database has found. So when you have your own sequence from your research, you may not have 100% percentage identity here, but you may have, for example, 97%, 96%. So they will be your significant results. So you will always check the percentage identity, your query coverage, and your e-value when you are looking at the blast results. The e-value should be as small as possible, whereas the percentage identity and query coverage should be as high as possible. And you generally consider 97% query coverage and 97% percentage identity, greater than these as the significant ones. So if you move down, so these are actually in the in the reverse order. The ones which are the most similar are presented at the top, and the ones that are a bit dissimilar are at the bottom. So if you see, this is 99.05%. So it means that 99% of the base pairs match to the sequence in the database, but few 1% slightly are do not match the the database sequence. So this is how you can get different results. So if we walk to the graphical summary, so here we have the this is what I was saying, the query coverage. For example, my sequence was 12,000 base pair long, and the search sequence was also to 12,000 base pair long. So it will be covering the entire length of my query sequence. But if it is not covering the entire length, there will be a small gap, and this gap will show you which of the sequences do not fully cover the query my query sequence. So this is the graphic summary, and if you click at any red dot, you can get the information about the hit that the database has found. For example, if I set it here, this will be Pseudomonas. It belongs to Pseudomonas category, but the species name is changed. But here we have the same Pseudomonas cutida that we took the sequence from, but the PLAS did not knew which species or which variant I took of Pseudomonas, but it has given me very accurate results. And if we move towards the alignment part, so this is a very important part, and from here you can identify the mutations as well. It gives you pair by pair, dot by dot, against each base pair. For example, this was my query sequence, and this was the sequence the database has searched across the database, and it matches each base pair to the each base pair of the search query. And this is of the first sequence, and we knew that it had 100% identity, so it will have no mutations. But here, if we go to it, okay, if we want to see the mutation, we have to set it like that. For example, here it is set as pairwise. I'm going to set it as pairwise with dot for identity. So what this one is going to do, it is going to put a dot between the two sequences. Okay, so what does that mean? The first one is 100% similar, so we do not we will not see any kind of mutation in that. The second one is also 100% identical, but here it was 99% identical. It meant that there was 1% mutation. So here you can see, if we had a G in our query sequence, it was replaced by a in the the subject or the sequence the database has found. So that is how you can find different kinds of mutations in your sequence that have occurred over time. So these might be different strains of the same species. They are pseudomonas, but you can see the species are different. So these have happened over evolution. These changes have happened over evolution. So these are the kind of mutations. So these are the kind of results you will get when you run BLAST, and you can select the one that has the top hit or the best match. And you can also have a taxonomy, and it is telling you the organism was pseudomonas and which group it belonged to, and which group it was the most similar to the pseudomonas putida group, but it was also similar to the pseudomonas monteli and different groups of pseudomonas. So the name of organism is same, but it has different variants or different species that it is similar to. So these are all the results that you get from uh, running blast on nucleotide sequence. So we're also going to do, okay, one thing here is one thing that you can also do with blast is, you might study these in your next classes, but you can also create a phylogenetic tree from here. You just have to click this button, and you can get the phylogenetic tree of your results, how your species that you have given is similar to all the other species of pseudomonas and all the other species in the databases, how it is similar to the others. So it gives you a kind of an evolutionary tree. So this is the one that we are trying to find, and these are the ones that it is the most similar to. And you can see that it is most similar to the pseudomonas here as well, so because it originally belonged to the pseudomonas category, so that is why. So now we are gonna move towards the blast P. That is the second part of our today's demonstration. The basic criteria of the basic working of all kinds of all variants of blast is typically same. The results are almost the same, but it is just the way how they work. Here you give an amino acid sequence and you'll get an amino acid result as a result. But in case of blast, and you give a nucleotide sequence and you give results as nucleotides as well. So from, we again gonna go to NCBI, and here now we're gonna select protein, because we want the protein sequence, not the nucleotide sequence. So here we have protein, and let's search P53 protein in humans. Okay, here we have it. We want its nucleotide sequence. Oh, sorry, we want it FASTA sequence. I hope it gives the correct one. Yeah, so this is the amino acid sequence of this protein. In the similar way, we are gonna copy it and we're gonna paste it here, and we can give a job title of the same the species. And you can see here now this is changed. This first it was the anti nucleotide, now it is going to search it against a protein database, and we're going to use a blast p because we are doing protein blast. And similarly we're going to hit the blast button. So this is also going to take a few seconds, and then you will have the results. Okay, so longer the sequence, the more time blast takes, but now we have a smaller sequence, so it does not take too much time. So this is the length of our amino acids length 393, the query length, and these are the metadata of the summary of the tumor protein P53. And similarly, you can set the percentage identity, e-value, and query coverage, and similarly, you will have the results here. So if you see here, the query coverage and percentage identity a little bit low than we observe in nucleotide, and this is because the protein to protein, conversion is a bit, different than the nucleotide one. So you may observe a different kind of query coverage and percentage identity here, but still above 90% percentage identity, high query coverage, and a lower e-value, it represents significant results. And similarly, you can go towards the graphic summary of the P53, and these are the results similar to the nucleotide one. And these are the similar alignments. And if you want to see the mutations, you can go for this one, and you can see which proteins have been changed to which protein over evolution. For example, if I had the P53 of human and this is P53 isoform of X1, so how those are being mutated over evolution. And similarly, we can have the taxonomy, where it belongs to, which kind of organisms have that kind of sequence in them. So this is the kind of results that you get at from blast. And from this as well, you can do your distance trees or your multiple sequence alignment. You have different kind of tools for this, but you can do it from blast as well, and you can get the similar trees from here as well. And if you want to do, for example, let's try T-BLAST N, that searches the protein against the nucleotide base. So I have the protein sequence and I want to search it across the nucleotide database. So this is how I am going to do it. I'll just give you an example. I'm taking the same tumor protein P53 from Homo sapiens, and I am going to hit blast by keeping everything else default. So now it is searching my protein against the nucleotide bases, and my result will be in nucleotides, not in protein format. So this is the basic difference between different variants of BLAST, and you should know which one you have to use depending upon your research question and what kind of sequence you have. So this is taking a little bit more time because it has to first convert the protein into nucleotide sequences, and then it is going to search it. Okay. So these are the results. Okay, we do not have the results right now. You guys are gonna hang for a minute or two. Okay. So these are the results, and you see here the personal identity is a little bit slow because it has given us the p53 tumor, but it is low because when you change a protein sequence into nucleotide sequence, the same protein can be encoded by different nucleotides. So that's why you will have a little low percentage value here, but the results will be similar. And if you want to see the alignments, you can see it from here. So these are all the kind of results that you Yes, so you can see here it converts the amino acid first into the protein into amino acid, and then it searches the nucleotide databases and search for the possible matches. So this is all from my side. Sorry. Okay. So let's have some questions now.
Thank you so much for your insightful and nice presentation as well as hands-on training. I hope our participants got the basic information as well as practical training what they needed to know about the blast and bioinformatics. Now let's move on to for Q&A. You can write the question in the chat box and I'm gonna take the questions for the answer. The first question we have that fast TQ is commonly used for next generation sequencing data because it provides information about the reliability and accuracy for the sequence. Is it true or what do you think about it?
Yes, fast Q is very different from fast A. It's not that much different, but it also has the the quality scores. So fast Q is a little bit different from fast A sequence. Fast A is basically the sequence of the nucleotides, and in fast Q you have four lines. The first line is your metadata, the second line is your sequence, third line is additional information, and the fourth line is quality scores. Because when you are doing NGS analysis, you have to get the quality scores, and the quality depends on the sequence confidence and how confident your sequence tool is about the correctness of that sequence. So that is why you use a fast Q sequence rather than fast A sequence for that.
Okay, thank you. The another question we have is if sequence alignment is dependent entirely. Sorry, in the moment. Okay, the question we have is if sequence alignment depends entirely on scoring matrices and gap penalties, can alignment ever be considered an object res- representation of evolutionary history? Miss Maniha Small?
Sorry, my mic was muted. I was speaking and my mic was muted. So
It's
I was
fine.
saying there it is not possible that a single sequence alignment is not 100% objective, but it's the best model we have so far for evolutionary history if you use it carefully. Because I already told you it is a heuristic approach. It used cameras, and you have to do a trade-off between the correctness and the accuracy and the speed. So we have to change the alignments depending on the results we want. For example, the scoring metrics, if we talk about the scoring metrics, uh, for example, PAM 2250 gives different scores for leucine and isoleucine, and if we change the matrix, it will change the alignment as well. And gap penalties as well, if they are open, they are extend, they will have different values. So two people can run blast on the STOM2 sequences and they can get a different alignment. So this part is a bit subjective as well. But if we talk about evolution, it is objective. So we cannot say that blast results are totally objective or they are totally correct, but this is the best we have so far.
Okay, I hope the participant got her answer. Then another question we have here is what are the limitations of using BLAST for sequence analysis?
Okay, so we do not use BLAST for sequence analysis typically, but we use it to search the matches against other databases. So I have already told you that we have to do a trade off between the speed and the accuracy. But because if we want accurate results, we would not get them in a very good form, in very easily, because for that we have to select the camera size very small. But if we want speedily results, then we use the camera size of large, that would affect our results as well. So this is one of the limitation of that.
Okay, thank you, Miss. The another question we have is, if I want to check a gene exists in catfish, which blast tool or database should I pick?
So that depends on if you have a DNA sequence or if you have a gene, then you will use BLAST N. But if you have a protein sequence, example from UniProt, then you can use BLAST P. But if you have a protein sequence and you want to see the DNA sequence, then you will be using tBLAST N. So it totally depends upon the kind of sequence you have.
Yep, thank you, Miss, for answering the question as well. The another question which we have, which is, when should we change the default BLOSUM62 matrix to BLOSUM45 or PAM to 250?
Okay, so Blossom 62 is a default, but you can switch it when you expect distant or close relatives. So BM62 actually means a 62% identity. So it is a default that has been selected by Blast, but you can increase when you want very close relatives to your species, and you can lower it. For example, you can set it at Blossom 45 when you want distant homologs. So that is how you can change the settings of Blossoms yourself as well.
Okay. Then we have another question is, um, why can two identical blast alignments have different e-values?
Okay, so two sequences can have different e values because that totally depends on the randomness. It's a random value. It searches the database and it searches if there is a random hit. So, two similar sequences, I have already told you that it is a heuristic approach. So, the e value or the percentage identity or query sequence, they can differ for the same sequences as well if you do it more than one times.
Okay. The another question we have is, can this a statistical framework underlying e-values ever fully capture the non-random structured nature of biological sequence? Or is there a fundamental mismatch between the model and reality?
Sorry, can you repeat the question? I didn't get it. Can the statistical framework?
Can the statistical framework underlying e-values ever fully capture the non-random structure nature of biological sequence? Or is there a fundamental mismatch between the model and reality?
Okay, okay. Okay, so no, e-value, statistical value can never capture the complete biology. This is a fundamental mismatch because, but if the model is still useful, because if you know where it breaks, because if there is a mismatch, there are different kind of assumptions for it. Sequences may be random, each site may be evolved independently, or gaps may be random. So it does not depend that the e value captures the total biology, but we still use e value because it is a relative measure. It is not an absolute truth, but it is still better than all the other ones we have right now. So we use it, that's why.
Okay, then we have another question is this step is necessary really because I completed my research project but didn't use blast in my photochemical in silico project of bioinformatics. So can you clear it?
Okay, no, it is not completely necessary because it all depends on your research question. If your research question is completely different and you do not, it is only necessary if you have run, for example, you have done a research such that and you want to find which species it belongs to, only then you run the blast. But if you have any other research question that could be, answered without doing blast, so you can do it as well.
Okay, the another question we have that if I have a DNA sequence but want to find protein matches, should I use BLAST X or T BLAST N?
If you have a DNA sequence that is not translated before, because there is a difference between complementary DNA and DNA. So if you have only a DNA, then you are going to be using BLAST X. But if you have a complementary DNA and you want to convert it into protein, then you will be using the later one.
Okay, the another question we have that is in this seeding stage, how is the K more size chosen? Does it differ for plus T plus N or versus plus P?
Okay, so yes, the default size does change with Blast N and Blast B. The default in Blast N is 11 and in Blast B is 6, but you can set it yourself as well. If you keep the gamer size large, then Blast is going to take a lot of time, because if you have a large gamer size, it is going to take time to match that exact gamer with the database. And if you have a small gamer size, it will search it quickly, but you will then be doing a trade-off between accuracy, because if you have a small value of gamer, it will search it very quickly, but it would match with multiple strains at the same time, so that might not be very accurate. So you can set your k value, gamer value yourself, but it all depends on the what you are trying to do. If you want to do a trade-off between accuracy and speed, then you can do the select the gamer yourself. If you want highly accurate results, you can select the high gamer value, but the default one for the blast is 11 for blast n and 6 for blast b.
Thank you. Now we have another question which is BLAST X is actually used for what? And what is the word for BLAST N as well? T BLAST N.
Okay, so T-blast N is basically used if you have a complementary DNA. For example, when you do PCR, you convert your RNA into into complementary DNA. So the result coming as will be a complementary DNA. So at that time, you will be using T-blast N. I'm sorry, the T-blast X. And T-blast N is used when you have a protein sequence and you want to search it for the nucleotide sequence. For example, if you have a protein sequence but you do not know the exact nucleotide sequence it matches to, then you will use T-blast N.
Okay, we have another question which is for the pseudomonas who did a result with e-value zero point 0.0 and 100% identity. Does that mean it's the exact same strain or could it be still different one?
Uh, it depends upon the query sequence and the length you have given. For example, if you have given a very short sequence, for example 100 base pair in the beginning of one species, for example Pseudomonas putida, but it might match 100% to some other species of pseudomonas as well. So it all depends upon the query length you have given. If you have given a large query length, and then it is exactly matching to the the sequence you have gotten in the results, then it will be the exactly same one. But it might be different if you have given the query sequence of very less base pairs.
Okay, now we have another question, I guess. How do you know that the mutations are due to change of species, or are those mutations affecting the function of that organisms?
Okay, so from BLAST, you cannot tell why the mutation is helping. Just tells you that this specific base is different from what it found in the database. But to prove why that mutation has occurred, you have to go to different other tools, and you have to identify. You may want to do variant calling to identify why the mutations have occurred, and the results that BLAST gives are not even 100% correct. So you cannot even confirm that if a database it is suggesting it has a mutation or a different base in the query sequence, but and a different one in the sequence it has given. So it might not be a mutation at all. So you would need different types of tool to confirm that it even is a mutation, and if it is, why is that mutation occurring. So BLAST only tells you the differences between the two sequences. It does not tell you the exact reason why that mutation is happening.
Okay, thank you, Miss. We do not have any questions now. We can go for that. Thank you so much, Ms. Mani Hashmal, for your insightful session as well as practical hands on training. Your knowledge, practical experience, and valuable guidance provided an excellent foundation for our participants. Now, to all participants, thank you for your active participations and enthusiasm. We look forward to see you in our upcoming sessions as well. Assalamu alaikum, everyone. Stay healthy, stay motivated, and keep exploring the world of science.
Thank you so much, Malika Ajmal, for a wonderful presentation. And I really like today's presentation, practical demonstration about the blast. And I hope it really helpful for the life sciences research leaders from all over the world. And thank you so much, everyone, for joining third lecture about the blast. And keep the learning, be consistent, and hopefully you will learn, and you will at the end you will be able to build your own project in the domain of molecular biology and bioinformatics. And it will be a wonderful journey, internship journey for you, for all the students. Thank you so much for your cooperation, and wish you good luck. And now, Maliha Ajmal, as it was the last lecture from your side, so if you want to say anything to the participants, floor for you, you can continue, and then we will take break. Thank you.
Okay, no, thank you so much. It was a very good experience for me as well, and it was an amazing experience to see such motivated people. And I wish everyone all the best in the future and lots of prayers for all. Thank you so much.
Nurpur, please conclude it. Thank you.
Thank you, everyone, for joining with us, and thank you, Miss Mani Hashmal. It was great. sharing the same stage with you and sharing the knowledge, your experience, and it was amazing. As well as the slides were also nicely organized. We pray for your future brightness, and that was all for it. I hope our participants also took their attendance through the QR code. And right now, we are going to conclude it. Allah Hafiz, everyone. Have a great night.